Hi, my name is Evan Flack, and I'm here with the 2013 Daniel I.C. Wang Award winner, Terry Papoutsakis. Welcome, and congratulations on your award.
Thank you.
So I guess just first thing I want to ask you is, what is the significance of this award to you? Just talk about kind of the influence Danny Wang has had on your research and his influence in the field of biotechnology.
So actually, I became aware of Danny Wang's existence while I was still a graduate student. And then in the early 80s, there was a series of conferences by chemical engineering in New Hampshire. And he used to bring his whole group with a van from MIT. So I was aware of the big activity and what he was doing. And it was a time where biochemical engineering was transitioning from the more traditional formative years of the transport phenomena into more new biology, molecular biology, more sophisticated models. The cell culture engineering was coming, into play. So I think Danny Wang sort of was there at that transition, and he defined it to a large extent, especially in the cell culture arena. And he was a very strong promoter of biochemical engineering as a discipline within chemical engineering, because that's where his roots were. And he was the one who insisted that there will be a marriage between biology and engineering. I think that was really the influence that shaped where biochemical engineering, which eventually became biomolecular, is right now.
And it was great that he was able to join us at your talk.
Absolutely.
It definitely
Absolutely.
Really, just his reception and everything
Yes.
really showed how
Yes.
significant of
Yes, and I was actually quite impressed that he made the effort, given his health state, that to make it to the meeting, and him and his wife, and it was really great to have him. I think it influenced, at least from my point of view, how this was done. His presence was very profound. As I was speaking, just in fact, when I found out he was coming, I even changed my talk to make sure that his presence, was taken into account for that.
Mm-hmm. Yeah. So tell us a little bit about what you talked about.
So I decided to talk about the work we do over many years on working with hematopoietic stem cells, the blood-making stem cells that seem to be, of course, a very important part of the technological developments and applications. And it's a new area that we got in the last 5 or 6 years, is to try to see whether we can use those cells to produce megakaryocytes. These are the very big cells that give rise, when they fall apart, into platelets, because platelets are a very difficult product. We cannot freeze them, and it's always short supply. So there's still a dream by a lot of hematologists and technologists to see, can we really produce platelets in a factory as opposed to relying on transfusions. So that's how the work started, and then we're trying to bring it into physiological conditions like the biomechanical forces, which comes out to be an important element in the biogenesis, the formation of platelets. So that's what my lab has been doing in the last 2 or 3 years. And we, by somewhat accident, stumbled upon some very small cell fragments, which in the beginning we thought it was junk debris, but it comes out they're very active components. So this is a new field in biology and bioengineering, the microparticles, because these particles really that come from each individual cell type separately, they're very potent because they recognize only specific targets, and they can be used to change the properties of the cells of the target. And now we think we can just take them and engineer them to be able to deliver molecules like DNA and RNA, proteins and so on, in the context of gene therapy, cell therapy, and also in the context of being able to collect those cells from each and every patient to have a patient-specific type of treatment, if you wish. So I think it's in a very exciting field because it's different. It's not a cell, it's not a protein, it's not a nucleic acid, but it's a combination of all of those, which makes it sort of like an exciting new field in biology.
Mm-hmm. Yeah. So you also just received a grant from ARPA-E to do some work.
Yes.
Can you talk a little bit about what that was?
Yeah, this is actually, we're negotiating the contract actually next week. This is a new field that ARPA-E is opening. There is an enormous abundance, as you know, of natural gas in the United States, enough to sustain the economy for more than 100 years, if not much longer than that. The problem with the natural gas is that you cannot use it immediately as a fuel for vehicles. So you're always looking for a liquid fuel. So there is an initiative now to say is, can we utilize, even especially locally in small facilities, technologies to convert the methane from the natural gas into a liquid fuel that will sustain and grow local economies and provide transportation fuel. So it's a very high risk type of new technology. They awarded, I think, about 12 or 14 different kinds of awards. Working with my colleague Wilfred Chen, they combined actually two different proposals. We had a full funded proposal and another one which is partially funded to allow us to develop new technologies that will take natural gas and convert it into liquid fuel. So this is, of course, a very fast track, very intense kind of a program. You have 3 years, a lot of money. We're going to hire a lot of postdocs and graduate students and work also with industry to really try to see whether we can develop a transformative technology to take advantage of this abundance of carbon, which also will mitigate environmental issues instead of releasing methane, which really is a very potent, I guess, atmosphere warming gas, temperature raising gas. So that's really very exciting to see that we can try the best we can with this opportunity.
Yeah, I mean, both of these projects sound like really exciting work, and we're looking forward to seeing some of the results over the next few years.
Of course.
Best of luck. Thank you again for joining us today. And
it's a real pleasure.
Congratulations on
Thank
the
you.
Wong
Appreciate
Award
it
this year.
Thank you.